Hello everyone. In this video we are going to talk about the digestion and absorption of proteins. We had already talked about the digestion and absorption of carbohydrates. Now in this video we are going to talk about the proteins. Uh, let's see. So we shall start about what is proteins, or what we call the polypeptides. Right. So dietary proteins are chemically long chains of amino acids bound together by peptide linkages. Right. So now, polypeptides proteins are peptides containing many amino acids, typically anything between 10 to 100 amino acids. Now, proteins are actually many amino acids linked together by peptide linkages. Amino acids linked by peptide bonds. Right. Sorry. Amino acids linked by peptide bonds. Peptide bonds. Right, so that is the proteins. So let's see, every amino acid has a amino group, a carboxyl group, and a R group. Right, so this is the alpha carbon to which all these four groups are connected. There are 20 kind of essential amino acids known in the nature, and we'll study about them in another video, but here we are going to study about this digestion of protein. So here we can see how a protein or a peptide linkage is formed. So this is one amino acid and this is another amino acid. The OH of the carboxyl group of one amino acid, the H of the NH2 group of another amino acid come together and a dehydration occurs. It is OH plus H, water is released, dehydration occurs, and a peptide linkage between CO group and NH group is formed. This is the peptide bond. Peptide bond is formed by the dehydration. Now, in just the opposite manner, the digestion of proteins occur. This is how protein or peptide linkage is formed. Now, how this protein or peptide linkage is broken, that we will see. So here we can see that this is the peptide bond, and hydrolysis occurs by dehydrolysis. Peptide bonds were formed. Now by hydrolysis, addition of water, digestion is occurring. Right, so OH from the water is added to here, and H is added to this NH2 group, and two amino acids are separated and the peptide linkage is broken. This is the digestion of proteins in chemical formation. So how this happens in our stomach? So pepsin is an important peptic enzyme of the stomach, which is active at 2 to 3 pH and is inactive at pH of about 5. So this pepsin enzyme is released in the stomach, is found in the stomach, and it is an important enzyme which is active at 2 to 3 pH and inactive at pH above 5. Now consequently, for this enzyme to cause digestion of protein, the stomach must be acidic. Right, so for the pepsin to work upon the proteins, the acid, there must be acidic surrounding in the stomach. Now the gastric glands secrete a large quantity of hydrochloric acid. This hydrochloric acid is secreted by parietal or oxygenetic cells in the glands at pH of 0.8. So there are oxygenetic, or what we call the parietal cells of the stomach, secretes HCl. Oxygenetic cells secrete HCl, hydrochloric acid, which makes the acid which makes the stomach acidic. Now, by the time it is mixed with the stomach contents and by secretion from the non-oxygenated glandular cells of the stomach, the pH average is around 2 to 3, highly favorable range for acidity for pepsin activity. Right. Let's go to the next slide. Sorry. So this is the figure by which I can very easily explain. So this is the stomach cells, and these are the parietal cells in purple. The rest are chief cells. So the parietal cells secrete the HCl, like I said, parietal or oxygenetic cells, also known as oxygenetic cells. So these parietal or purple cells secrete the HCl, hydrochloric acid, right, and that makes the stomach acidic. And these chief cells, all these chief cells, they secrete the pepsinogen enzyme, the inactive form of pepsin known as pepsinogen. It is inactive right now, so known as pepsinogen. So this is inactive, and then HCl acts on a pepsinogen. HCl acts on pepsinogen, and the pepsinogen is converted to pepsin. Pepsinogen is converted to pepsin, the active form, by the acid, right? So then pepsin digests the protein into smaller peptide fragments. This is a whole protein, and the pepsin acts on it and breaks the protein down into smaller peptide fragments. Now, of the important feature of pepsin digestion, it is ability to digest the protein collagen that is affected little by other digestive enzyme. Collagen is a major constituent of intracellular connective tissue at meat. Therefore, for the digestive enzyme to penetrate, needs to digest the other meat protein, the necessity to collagen fiber to be digested. So pepsin, I'm sorry for the grammatical mistake in the paragraph, I don't know how it happened. So pepsin enzyme could digest collagen, a kind of protein that is present in meats. Let's go to the next slide. So pepsin only initiate the process of protein digestion, usually providing only 10 to 20% of the total protein digestion to convert the protein to proteases, peptones, and a few polypeptides. So the protein digestion, unlike carbohydrate digestion which starts in mouth with alpha amylase, the protein digestion starts in the stomach, right here in the stomach. So the first enzyme, that protein is digested by pepsin, which is found in the stomach, and only 10 to 20% of protein digestion occur in the stomach, right? So what happens? Pepsin digests the proteins and converts into proteoses, peptones, and polypeptides, right? So only 10 to 20% of total digestion occurs in the stomach, while the rest of the digestion of proteins happens in the small intestine. Now, the splitting of proteins occurs as a result of hydrolysis at the peptide linkages between amino acids. So, I already told you what is the digestion of protein. It is the breaking of the peptide linkages between two amino acids by the process of hydrolysis, right? So, hydrolysis occurs of the peptide bonds, and that's what digestion is. Let's go to the next slide. So most protein digestion results from the action of pancreatic proteolytic enzymes. So we already know that in stomach only 10 to 20% of the digestion of protein occurs. The rest, most of the protein digestion occurs by the pancreatic proteolytic enzyme, right? This is the main place where the protein digestion occurs. So most protein digestion occurs in the upper small intestine, in the duodenum and the jejunum. The upper part of the small intestine are called duodenum and jejunum, while the lower part is called ileum. Now the duodenum and jejunum is the place where most of the protein digestion occurs. Under the influence of proteolytic enzymes from pancreatic secretion, immediately on entering the small intestine from the stomach, the partial breakdown of products of protein food are attacked by the major producer lytic pancreatic enzyme trypsin, chymotrypsin, carboxypolypeptidase, and elastase. So, the major pancreatic enzymes which helps in protein digestion are trypsin, chymotrypsin, carboxypolypeptidase, and elastase. Right. So these are the four major enzymes coming out of the pancreas to digest the protein. Now, activation of pancreatic enzyme. Now we already studied that for the pepsin of stomach to get activated from pepsinogen to pepsin, they need acidic medium for which HCl is secreted. But for the activation of pancreatic enzyme, a basic medium is required, right? So now enteropeptidase activates trypsinogen. It converts trypsinogen inactive into trypsin, which is active. Now trypsin then activates all the other pancreatic zymogens, including itself. So enteropeptidase is an enzyme synthesized by and present on the luminal surface of the intestinal mucosal cells, the brush border membrane. So let's see this. This is an important part. So what happens? Enteropeptidase. This is enzyme required to activate trypsinogen. Now when trypsinogen is activated and converted to trypsin, right? And trypsin, this active trypsin then activates all the other pancreatic enzyme, which includes itself and the chymotrypsins and elastases. All the other enzymes are in fact activated by this trypsin. So enteropeptidase is an enzyme synthesized by and present the luminal surface of intestinal mucosal cells. Let's go to the next slide now. So this is a revision kind of slide. In the stomach, what happens? The parietal cells, the oxygenatic cells secrete HCl, chief cells secrete pepsinogen. Now pepsinogen in the presence of HCl is converted to pepsin, which is the active form, and is then the protein is broken down into protease with acidic and aromatic amino acid. Now exocrine pancreas secretion in small intestine. Trypsinogen requires enteropeptidase enzyme to convert it into trypsin. Chymotrypsinogen is converted to chymotrypsin, the active form, by the action of trypsin. Then proteoelastase is converted into elastase by the trypsin, and procarboxypeptidases is converted to carboxypeptidases by the action of trypsin. Right. So enteropeptidase, like I said before, enteropeptidase activates trypsinogen to trypsin, and then this trypsin activates all the other enzymes, chymotrypsin, proteolastase, and procarboxypeptidases. So it is an important part to remember. Let's go to the next slide. Now trypsin and chymotrypsin split protein molecules into small polypeptides. So trypsin and chymotrypsins split protein molecules into small polypeptides. Now carboxypolypeptidase then cleaves individual amino acids from the carboxyl end to the polypeptides. Okay, and proteoelastase is converted into elastase, which then digests elastin fibers. So all have different functions. Like I said, trypsin and chymotrypsin does the same function of splitting the protein molecules into small polypeptides. Carboxypeptidase then converts these polypeptides into amino acids. And proteoelastase is an enzyme which is converted into elastase, and this enzyme elastase digests elastin fibers. So let's see what we studied already, that in the stomach, if this you suppose is the stomach, so here is the pepsin, which digests only 10 to 20% of the protein and requires HCl for the digestion, right? And then the food goes into the smaller intestine, where there is pancreas, which secretes trypsin, chymotrypsin, trypsin, and elastase enzyme, which are all activated by enteropeptidase. Enteropeptidase. This is an enzyme which is synthesized in the small intestine, and it activates trypsin, and then trypsin activates the rest of them. Right, let's go to the next slide. So here is this figure which may help you remember. The stomach secretes enzyme pepsin. Action of pepsin is to digest the protein into proteoses, peptones and polypeptidates. Only 10 to 20% of protein digestion occur in the stomach. The second is the small intestine, where there are 4 important enzymes secreted: trypsin, chymotrypsin, carboxypolypeptidase and proteolastase, secreted by the pancreas. And the action of these enzymes, the trypsin and chymotrypsin, split the protein into small polypeptides, while the carboxypolypeptides peptidase digest the small polypeptides into amino acids. Right, so this is their action. Then they go to the aminopeptidase and dipeptidases are the enzymes we are going to study about later, and their actions. Right, so we will come back to this figure later. Right, for now this is what you need to remember up to this. So let's come to this slide. So here, digestion of peptides by peptidases in the enterocyte that line the small intestine villi. So up to the pancreatic secretion we had already read. Now we are going to read about the digestion that occurs in the intestinal villi. We had already studied about the intestinal villi in the carbohydrate digestion part, that the small intestine lined by thousands and thousands of villi, and they are something like this, finger like projection, and every villi has millions of other small projection known as microvilli. Right, and these microvilli have the cell called enterocytes. These enterocytes cells are there. So if you're not clear on this, go back to my earlier video of digestion of carbohydrates and see that before studying this, so you know all about enterocytes. Right, so let's start. So how digestion occurs in the enterocytes or intestinal villi? The last digestive stage of that proteins in the intestinal lumen is achieved by internal sites that line the villi of the small intestine, mainly in the duodenum and jejunum. These cells have a brush border. We are talking about the enterocyte cells. They have a brush border that consists of 100 of microvilli projecting from the surface of each cell. In the membrane of each of these microvilli are multiple peptidases. These enterocyte cells present in the microvilli have many enzymes known as the peptidases that protrude through the membrane to exterior, where they come in contact with the intestinal fluids. Now, I am sorry, there are so many grammatical mistakes. So, 2 types of peptidases enzyme are specifically important: aminopolypeptidase and dipeptidases. So let's see this figure and figure this out. So what happens? So there are amino acids digested by the pancreatic juices. There are large polypeptidase, and there are dipeptides and tripeptides. So what happens when they reach this enterocyte of the microvilli, enterocyte cell of microvilli, so this is, and they have this brush border, finger like projection brush border. So this brush border have many enzymes called the dipeptides and tripeptides. Right, so when at this brush border, when these amino acid or large polypeptidase come here, these dipeptides and tripeptide and peptides enzyme are converted into amino acids. Right, or large peptides are also converted into small dipeptides and tripeptides. So this is where most of the digestion of protein occurs. Now the aminopolypeptidase and dipeptidases split the remaining larger polypeptides into tripeptides and dipeptides and a few amino acids. So there are 2 enzymes, like I said, aminopolypeptidase polypeptidase and dipeptidase is at the brush border of the microvilli, and their only function is to break down the larger polypeptides which come into contact with them in the microvilli. These enzymes come into contact with the polypeptides and break them down into small amino acids, singular amino acids, or small dipeptides and tripeptides. So no bigger molecule, no bigger protein molecules remain in the small intestine, right? So this is the function of aminopolypeptidase and dipeptidase enzymes present in the brush border of the microvilli of enterocytes. Now the amino acid dipeptides and tripeptides are easily transported into the microvilli membrane into the interior of the enterocyte. Finally, inside the cytosol of internal site and multiple other peptidases that are specific for remaining type of linkages between amino acids within minutes. Virtually all the last dipeptides and tripeptides are digested to the final stage to form a single amino acid, which then pass into other side of the internal site and reaches into the blood. So that's all right. So the larger polypeptides are first broken down into amino acids, singular amino acid, or dipeptides and tripeptides by the brush border enzymes, amino polypeptidase and dipeptidases. So now only the dipeptides and tripeptides remain, right? I mean, three amino acids, sorry, like something like this remains: lysine, glycine, arginine. This is a tripeptide, and like glycine, arginine, this is this is a dipeptide. So only like these dipeptides and tripeptides remain. All are broken down into such kind of dipeptides and tripeptides. And then there are other enzymes within the enterocyte cell. Within, if this is enterocyte, there are other peptidases which breaks down these dipeptides and tripeptides into singular amino acids. Like there will be only glycine, only arginine, and there will be only lysine, right? So they'll all be separated by the end of the digestion when they enter into the enterocyte, right? So let's figure this out. So proteins, there's a whole protein. Pepsin acts in the stomach and converts them into proteoses, or digests them, breaks them into proteoses, peptones, polypeptides, small polypeptides, and then they go through the small intestine where there's trypsin, chymotrypsin, carboxypolypeptidases, proteolastases, and are converted to smaller polypeptides and amino acids. Then peptidases are the enzymes that work on them, and they are then converted into singular amino acids, and no polypeptides or peptide chains remain, and the final digestion of proteins had occurred, right? So thank you for watching. In the next video we are going to talk about the digestion of absorption of these protein. We have already studied about the digestion.